We would like to show our respects and acknowledge the Bedegal people who are the traditional custodians of the land on which this meeting takes place, and to elders past and present.
Hi everyone, oh, it's very loud. So my name's Andre. So I am a field application scientist for Nanopore that's actually just started, maybe about 2 months ago. You don't need to worry about my title because I don't actually know what it means either. And so essentially what my role here is I'm just meant to help out if you've got problems with Nanopore or want to come up with projects. I'm here to help guide you, so you can always contact me for that. Just a little bit about myself. I have a background in transcriptomics, RNA, but now, of course, I'm diversifying now to all the applications of Nanopore. So I'm hoping I'll get to meet more of you. I've met some of you already, and so hopefully over the coming months I can come to your labs and learn what you're doing and see where we can work together. So first up, I'm just going to give a little technical update on Nanopore. Can I get a show of hands who's used Nanopore before? Is it the majority here? Okay, oh, that's good. Oh, I'm glad to hear it. So just like how I'm brand new, we've also got a new device here called the Mark 1D. This is kind of our portable device that you can take out into the field. Maybe you want to go into the school, have a little project there. It's very small, streamlined, and takes our mini and flow cells. And what's improved in this device over our latest ones is that it's got better thermal control, and it's soon going to be compatible with an iPad. And when I say it's small, it can just fit in a pocket like this. Now, it works up to 35 degrees, so it's not going to work on our scorch of a weekend that we just had, but it's still going to be helping us in going out into the field. So now to move on to the more technological side. So this graph might be a bit scary, but if I was to have a show of hands, who would say that Nanopore is known for its highly accurate base cooling? Oh, yeah, so I'm not surprised. Now, I want to change that. So with our latest update, this is the first time we've actually updated the architecture in our base cooling system. So now we're up to Q26 with our super accurate base cooling, or SUP, as you may have heard of it. And that's a 1.5 increase. So for those that don't know what Q26 means, essentially that translates to about 99.75% accuracy. So if you sequence 1,000 base pairs, 998 of those approximately will be correct. So we're getting really close. I would say we have very nice, accurate base cooling, and that's at the individual molecule level, so not a consensus sequence. So you're going to feel it for every molecule you're sequencing. Now, more in my comfort area, this is also true for RNA. So for RNA, we've reached a Q of 19.4, and what I really want to emphasize on this plot is just 2 years ago, or 1 year ago, at the Nanopore community meeting in 2023, we've already had almost a 5-point increase in the Q score. So you can see we're rapidly approaching a Q score of Q20, meaning 99% accuracy. So we're getting very, very good sequencing. And what's really nice about this is that it's all backwards compatible. So because this is all done on the software side, if you've already got data that's sequenced on the RNA-004 pause, then you'll be able to rebase cool these and take advantage as the software improves. And you might be thinking, okay, a lot of you had your hands already. We've already got data. It's working fine. But what's the advantage of having better accurate sequencing? So the main advantage I find is that it gives you more confidence. If you're, for example, looking at SNPs or SNVs, you can kind of push the limits of how many reads you actually need to be certain that you see it here. So I've just pulled this paper off bioRxiv, which I thought was really pushing the limits. They're doing single-cell whole genome sequencing of individual neurons. So you can imagine they don't have a lot of DNA that they're working with. And what they're able to do is, with Nanopore, they were able to identify more SNVs compared to short-read sequencing. But in addition to that, they can phase, they get haplotype resolution, and as a bonus, they also can look at complex structural variants such as and TEs. We also, in a similar vein, for those working in single-cell RNA-seq, we've updated the workflow, so workflow single-cell version 3, and this also allows now for SNP analysis calling for your single-cell RNA-seq experiments. Something that's also very close to my heart is RNA modifications. I was just speaking to someone just before about that, just before I started talking. And what I want to talk about here is that now, well, previously with Minnow, you'll be able to get M6A calls for your RNA-seq expression, RNA runs. And now we've added 2-minnow pseudouridine. And then for those that use Dorado-based cooler, we've also added inosine and M5C. So we're expanding our repertoire of RNA modifications that you'll be able to detect in your experiments. And I think this is really exciting because it opens up more scientific questions. You're essentially adding layers of information that you're getting effectively for free if you're doing direct RNA sequencing. Here's an example that I pulled off where they used direct RNA sequencing on acute myeloid leukemia patients pre and post treatment. And they found that when they looked at the M5C in these patients, that it reduced after treatment, and this correlated with remission. And when they looked at the patients suffering the acute myeloid leukemia, the hypermethylated genes were linked to myeloid cell activation. So it shows that this extra information that you're getting can really enhance the biological significance from your data. And so just to round out the RNA section and what's coming in development. So I think the big thing that I think a lot of people are looking forward to is RNA multiplexing is in the works. So you'll be able to run more samples together, which will hopefully allow you to use less flow cells and get more information. We've got things like targeted RNA-seq. We're, of course, improving our base calling and increasing the repertoire of modified bases. So there's 170 plus of those. So we want to keep, we've only got 4, so that's a long way to go, but a lot of room and hopefully a lot of interesting insights. Just as another quick section, who here knows what adaptive sampling is? Okay, so a bit less than before, okay. So for those that don't know, adaptive sampling is something that's very unique to nanopore sequencing. Essentially, using the same libraries, you'll be able to enrich for target regions of your genome. And so what that means is that without having to do any PCR, without having to strip away the methylation, you can greatly improve the coverage over the areas that you care about. And you don't have to waste time sequencing areas that are not too informative for you. The way it works is you can imagine the pore here. Unfortunately, the animations were wiped out, but as the DNA is going through the pore, it's being real-time aligned to a genome. And then if it falls in the region that you're interested in, it will keep being sequenced. But if it doesn't, it's going to be rejected so that another strand can enter and sequence the parts that you're interested in, essentially resulting in an enrichment. Just as a little example here, so here we've got the RFC1 gene, and it's a targeted region. And you can see in this IGV plot, there's a lot of reads that are falling in this region, and in the off-target regions, it's a lot less. This shows how you can improve coverage of areas that you're interested in. And it's very simple. It's just a click of a button, and you provide a bid file. And we've also got some improvements coming for this, or have already come. So for those of you that have a P24, I don't think that would be many of you, but for those that do, now you can run it on all 24 positions without any major impact to your performance. And also, for those that do a lot of multiplexing, we used to have this barcode balancing feature. And so we've actually we're going to be updating that, it's not out yet, called sample until. And so the aim of this is to make sure you try and get as much or an equal amount of representation from all the barcodes that you have included. Maybe they didn't have the same quantification, or you weren't able to quantify it before you pulled them together. And the way it works is or however many barcodes you want will be sequenced, and you set an upper limit for how much you like. Then once the first barcode hits that upper limit, it's going to be stopped, and if the pore sees that barcode again, it's going to reject it and try and get another strand with another barcode, effectively trying to enrich for the ones that are underrepresented. Just my last slide will be the T2T assembly. So we are creating an assembly pipeline for this. And so essentially this is your telomere to telomere assemblies, which is a combination of ultra-long reads, pore-C, and the APK kit. So we have a bioinformatic pipeline that's coming that should make that a bit easier to do. And so you can find that all on the Nanopore community website as well. So with that, I'd like to thank everyone. And if you can find me, unfortunately I'm only here till 5 o'clock, so you won't be able to catch me today. But please contact me by email if you have any questions. It's andre.sim@nanoporetech.com, and I'll be happy to talk with you.
Thanks, Andre. Do you want to ask Andre some questions now? Or Mike? Mike is also here. He's the field application scientist from For all of Australia, right? You're the lead? Yeah. You should. I know there's no sales rep anymore in Sydney, but that was a great job. You could also become a sales representative. I had a question with the last slide. What's APK?
Assembly polishing kit.
Assembly polishing kit.
Glad he got that because I barely know.
There you go.
That's what it does is get through homopolymer region. You actually attach it like an MA6, which is synthetic, which is not a DNA. And so instead of just getting a flat line that we can't differentiate how many nucleotides are in a sequence, it gives it a signal, and it basically gives us a handle within that region. So now we can actually sequence all the way to the chromosomes or to the centromeres and through the homopolymer regions.
Does that affect the base calling of the non-homopolymeric regions much? Fascinating. Does anybody have a question? Robin? I'll bring the mic to you. You get to embarrassingly broadcast your voice in the whole building.
Thank you. One of the first slides you showed this graph where you had the SNVs and SVs and the percentage of accuracy and that it improved, right? For what kind of depth is that? Is it like any depth or only at certain specific sequencing depths?
So I don't want to, so this obviously isn't work that I did, but just to summarize from the paper, so they took individual neuron cells, so the sequencing depth is going to be a lot lower. So I think each individual cell, the coverage was only about 20% of the genome. But that was for both the short read and the long read, so they were about equivalent to each other. Yeah.
But you can give them recommended for us, 20x is structural variance.
If you're doing a proper full genome experiment where you've got a lot of DNA input, 20x would be the normal coverage that we would do for structural variants.
For SNPs and indels at 30x coverage, you get enough. You basically get all the clinvars and everything like this. So 30x is still the gold standard. We still want that, but when you talk about just structural variants and things like that, and methylation, structural variants and methylation, you get down to 10x for methylation and then 20x for structural variants. We are now the gold standard for methylation. We are actually about 99.6 at like 15, 20x of all the methylations, and if you look at other platforms that do bisulfite or something like that, what happens is they miss all the GC-rich regions as well. We can now look at methylation on a single molecule, single strand with 100% accuracy. And we get 5-HMC at the same time.
I think, Robin, you should have prefaced your question saying that Robin's been digging into comparing short read sequencing to long read sequencing for variant calling. And he has some interesting data that hopefully he'll get to share with us soon. Any other questions for the Nanopore crew? No? Really? Yeah? Well, if there's no more questions, we have a bit of an announcement. Thanks to Nanopore's support. By the way, Nanopore have subsidized the drinks and the food mostly subsidized it. So thank you. They're also supporting us for a competition. So we're going to launch I'll bring this up here, this Project Drive initiative where, can you see that? There we go. We're happy to take abstracts to win 4 flow cells worth of data. So you can submit an abstract for a small research project and get 4 free flow cells of data, which is about almost 6,000-7,000 Australian dollars worth of sequencing data. It's enough for 4 whole genomes and a ton of transcriptomes, I think. 1 month closing date for abstracts, give you a bit of time. We'll probably we might not have time to announce the winners for the next genomics seminar, but perhaps the following one will announce the winners. Main conditions are that you have samples that are ready to go soon, and that you cover some of the costs associated with quality control and the staff time at the center to prep the samples. But you have complete control over what you want to do with the data, and I'm quite interested. I think I might throw my hat in the ring if there's probably a bit of a conflict of interest there. But yeah, so please apply, share with your friends, or don't if you want to increase your chances of getting the award. Go ahead.
We just want to clarify that this is not for a pilot project where you're just trying to say, get some data to put in a grant. This is actually, you have samples in the freezers, and you sort of need that conversion. Do I want to switch from short read to long read, or I have a cohort that I'm already ready to sequence? And this is just to give you that proof that it's going to work for you with your samples. And we'll work with you, Andres here, myself will work with you to get the data, to generate the data, as well as process the data and interpret data to your tertiary analysis.
I think that in-kind support is value add in itself. Okay, on that note, let's move to our first scientific, well, thanks again, guys, and safe travels to Melbourne. Yeah, see you tomorrow. I'm heading up to Melbourne very early tomorrow. Our next speaker, it's my great joy to invite Dr. Mark Pinese to the stage. I've known Mark for a while. He's a fantastic scientist. Mark is a group leader at the Children's Cancer Institute just next door, but he lives in Brisbane, which is why we kind of rushed to get this seminar today, because Mark was in town, so we wanted to be able to share some of his work, and happy to have you as one of the speakers. With that, I'll hand the mic to you and get your slides up for you.
Thank you so much, and that's why I've got the beer here. Brisbaneite can't get me out of the city for too long. And actually, it's funny, Martin. Oh yeah, all good, thank you. Oh, isn't that mine? I'm going to need that later. When you was putting the slide up at the start, talking about these seminars not being, rebooting them, it made me realise that I gave my first Ramachodi linked seminar in 2005. So a few things have changed since then. I've got a lot fewer hairs, and also I think at the time I hadn't been in meetings since 9 a.m, but hopefully I'll make some sense to you today. So I'm going to be talking about our work on trying to understand why kids are getting cancer. And relevant to this topic and this audience really is how we decided to do some long-read sequencing, very, very slow to the party, but we made it there eventually, and how that actually really helped solve some pretty serious issues that we had, because although we're part of an enormous and well-funded program, we'd only done short-read up until this point. So before I get into it though, I just want, who works on cancer in here? Hands? Barely any. Good, I'm glad I have these slides then. Okay, so just to get everyone on the same page. So cancer, I think most would agree, is a genetic process that is really driven by the accumulation of what we call somatic mutations throughout the lifespan. So these are genetic changes that occur in your non-germline cells. And over time, they accumulate in certain cell populations and lineages, and eventually act to overwhelm cellular control processes, leading to uncontrolled replication and cancer. And completely consistent with this being an age-related process, we observe that the incidence of cancer increases with lifespan. And it's no surprise then, cancer is a disease of advancing age. And these are data from the Australian Institute of Health and Welfare, but you'd see the same pretty much in any cohort, that cancer occurs at a highest rate the older you get, and it actually fits really closely to a power law relationship. And on the basis of this, you'd expect that at the very youngest ages, in those people who've not lived long enough to accumulate these mutations, that you would observe the lowest rates of cancer. Of course, that's what you'd expect, but it's not what you see. And indeed, when you look at the very youngest people, that's where you observe an excess of cancer, a quite significant statistical excess. Now, why is this so? These children have not lived for long enough to accumulate somatic mutations, so where is this cancer coming from? And of course, the answer is that this is only part of the story, and that indeed these somatic mutations which are driving so many cancers do not work in isolation, but really occur in the context of the germline. You know, the genome that you're born with that modulates the rate at which you accumulate these mutations, but also can change your very biology and lead to syndromes which are effectively cancer risk syndromes. And so being focused on childhood cancer, that's really our research in my group. You know, what are these genetic changes in the germline that are driving kids' cancer, that may be putting children on the road to this very rare but potentially very devastating disease? How many kids with cancer actually were born with this risk, and could we have found them before they got cancer so that we could have the best chances of managing it effectively. So these are the questions that we aim to answer. This is my team. I won't go into all of them, but just to say that we're a kind of mixed team, clinical, lab work, dry lab. But pretty much all the work I'll be talking about today is done by Naomi, very talented RA in my team, and I'm really just talking through my hat on this one. So all credit goes to Naomi, all fault goes to me. And so we try to understand genomically what is driving kids' cancer, and one of the big resources that we use for this is the ZERO program. So this is a very large precision medicine program that we're part of. It's now funded to the tune of just under $200 million, so we have quite a lot of funds to work with. But as part of that, we have data for over 2,000 patients, primarily whole genome sequencing based on short read sequencing. And this is where I think you can probably all see we're going. So I wear a second hat, not only as a lab group head at CCI, but I also co-lead this part of ZERO. So if you're interested in this, I'm happy to answer some questions about it later, because these data are accessible for people to use. And so one of the simple things that we do with all these data is we just say, okay, how many kids who walk into the clinic diagnosed with cancer have a genetic risk in their genomes that we know about? And so this is very simple. We know which genes to look in. We know which kind of variants to look for. We do short read sequencing. We find the variants. And we find that in 16% of kids, we find some pretty clear reason for why that child might have had cancer in their germline. These are things they were born with. But, and there's a pretty big but, there's a big asterisk next to that number. And that asterisk is because these are SNVs and indels only, found by short read sequencing only in coding regions, only in genes that we already associate with cancer, and only ones that are very clearly pathogenic. So there's a lot here that we could be missing. And so the question becomes, in this 84% of patients for whom we don't currently report a risk back to the clinicians and the families, could there be something in there that we're missing? And this is very important, because if we could find those kids who have a cancer risk that we don't currently understand, we can report it. That changes how we manage their cancer today, and it changes how we manage risk in the entire family, because they all may share this risk and they all may be at risk of cancer. And we do indeed have families where, 2 or 3 kids in the family have had kids' cancer. It does happen. So the question that we asked was, all right, what's in that 84%? And how often is kids' cancer due to complex variation that we don't currently examine? Partly because we can't, partly because it's too hard. And so I tasked Naomi to look at this, and we thought one of the simple places to look, simple, was in transposable elements or mobile element insertions. So the idea that perhaps this is causing some genes to be disrupted and this is creating cancer in kids. So what are these mobile element insertions? Many of you probably heard about them, jumping genes, transposons. But in humans, we're talking about primarily 3 different classes of DNA sequences which have the ability, through one way or another, various mechanisms, to insert themselves into pieces of DNA that they don't normally belong in. And when this happens, it's not hard to see. Some of these things are 6 kb long. That's going to disrupt gene function, potentially lead to a serious issue. And indeed, this is known as a cause of human disease. This is an example of a girl with orofaciodigital syndrome type 1, where you can see, so this is not a cancer syndrome, but a developmental one, where you can see right in this OFD1 gene, there's a clear insertion of something, not sure what, but something, right in the coding region of this gene, which is not in either parent. And indeed, in this case, this was the cause of her disease. So how often do we see that in our kids? So I thought, great, brilliant. That's the question. Off you go, Naomi. We found a tool that does it. We had a cohort which is part of ZERO called Predict. What's different about that is that we have parental samples as well, which can help to differentiate inheritance. And I thought, fantastic. Off you go, Naomi. 30x short read. We got the data. 228 patients. Run melt. We'll get a paper in a few months. Easy done. That was the plan. And she ran the tool. That worked out great. But what happened was we got inundated with rubbish. 251,000 mobile element insertions were found. That's more than 1,000 per patient. 573 in cancerous genes. So straight away I said, either we've got ourselves the paper of the century, or maybe it's not quite what we thought it was. And when we looked a bit closer, there were some patients who had 6 genes hit by an MEI in their genomes. That's probably not real. We often see 1 gene hit in a kid, sometimes 2 if they have a particularly severe syndrome, 6 never observed. And things got even more suspicious when we started looking at which genes got hit. And you can see a few here. This is RUNX1 right on the end. And we also have WT1, ALK. Almost every patient had one of these. So again, either the best finding that I've ever had in my career or rubbish. So it was indeed rubbish, and when we looked more closely, a lot of these things were not real. Naomi did a lot of work in trying to differentiate what was real and what wasn't. This is again short read. She did a lot of filtering. She used the parental samples to try to tease out what was real and what wasn't. And what I said is just go really aggressive on this. Filter out every bit of rubbish that you possibly could. Even if we find just a few, it's a start. And so she got it all the way down, 573 in cancer genes, filtered it down to 20 probably real ones, 3 that linked with the diagnosis, so they made sense in the clinical context. And I went, that's probably a bit too much filtering. So we had a quandary. We had a lot of data, we had a good question, and we were stuck. There's literally nothing we could do here. We did everything we could on the short read to try and figure out what was going on. But we ended up getting absolutely nowhere. And these were the kind of variants that we saw. So again, we had what looked like insertions in perfectly sensible genes. So this is a PAX5 insertion in a 2-year-old boy with B-ALL. These are linked. PAX5 is associated with blood cancers and blood development. It made sense. But can we take this to a diagnosis? Can we go any further? We couldn't. And this is where we were stuck. And this is where we happened to run into Martin at I think the one of the retreats, right?
Yeah, it was the Children Cancer.
At the kids, the cancer biology retreat. And Martin said, I can help you. And this was fantastic because we had no idea what we were doing. So what Martin proposed was adaptive long-read sequencing, just like we've heard, using Oxford Nanopore, to just target these regions that we found, sequence right through them, find these events, and just figure this thing out. This was a godsend because we had no idea what we were doing, but RAMA were fantastic. They worked with Naomi to identify the regions for this targeted sequencing, actually get it to work, because I wouldn't have the first idea how to actually get it to work myself. And full credit to RAMA as well, because we were going up to a conference late last year. I was getting a bit, really wanting this data to be ready, and they pulled out all the stops to get it done in time. So of course, 2-day turnaround to be expected for everyone, right, Martin? Okay, I'm never being invited again. But no, seriously, working with RAMA was a real pleasure for this project. And so how did it go? Well, it went really, really well, actually. So this is the PAX5 variant that you saw earlier. Again, we saw something in the short read, but we couldn't go any further with it. And straight out of the bat, the long read came in and said, actually, no, that's nothing. So as interesting as this looked on short read, once we got to long read, it went straight through it and said, look, that's a poly A expansion in an intron, no one cares. That's not a variant. And that was actually super helpful because looking at this, it looked far more interesting. You could see the poly A, but then, all of these clipped reads over here looked like there was something going on. Turns out, nothing. So that was super useful. A good, solid negative on that one. And we actually ended up seeing the same event multiple times. This wasn't picked up in the short read. It's there, you can see it in the data, but this is a different patient. Long read found it again, and when we went back to the short read, we were like, oh, we see that same pattern, except for some reason the caller didn't pick it the first time round. So again, really just reinforcing this is probably just a population polymorphism. But that said, we also found some fantastic things. So this is a 15-year-old girl with Hodgkin's lymphoma, and this is the ATM gene, just right at the start of an intron here. And again, something on the short read, but not really sure what it meant. And long read found an LU insertion, 315 bases. You could read right through the thing. It was there, clear as day. Didn't even need a caller. You could just see it. It was brilliant. And this is, yep, just really zooming in. And then we got to some really interesting ones. And this is one that I really wanted to show you because this was a case that actually went back to the clinic. So this is a 3-year-old girl with retinoblastoma. Retinoblastoma is an eye tumor, and it is one that is very strongly associated with a mutation in the gene RB1, and especially so when you have a family history. And this girl did have a family history. So we knew, looking at this, that there was probably something in this gene, but we could not find it, even though we looked. We knew where to look, and on the short read data, we were literally looking manually, couldn't find anything. And this is why. If you look at this, look at the mapping qualities around here. It's all absolute trash. And there are genes like this that we just can't do anything with in short read, and this is one of them. So we were looking around that region, and then this is one that we submitted for the long read as well. And it's a little less clear than the others, but right here, you can see there's clearly, based on the clip reads, something going on, and it turns out that this was not even an MEI. This is an endogenous long non-coding RNA that had been inserted into this gene. So it was from chromosome 14 being inserted into 13 on RB1 and predicted to affect splicing, and that was recapitulated on the transcription, on the transcriptome, and this led to being returned to the family as causative of that cancer. So this was a finding that there is no way we could have found it on short read. So not only is it in an unmappable region, but it was actually it was an insert that we didn't even know to look for. So really just emphasizing that you can find things that you wouldn't even expect to see. So I know I'm on time, but that was it in a really quick overview. If I had some conclusions, and for some reason my numbers are very small, Nanopore actually easily resolved several key mobile element insertions and weird SVs as well that you wouldn't expect in key cancerous genes. And the Ramachottis was really great to work with. Their adaptive seq was very easy for us. It was very cost effective too, so that was always nice. And they did it really quickly. Again, it was great. We just gave them the DNA, got back, mapped things, and Naomi went off and did it. And as someone who's done a lot of short read sequencing in my time, but zero long read, I was a little bit nervous about how it would go, and it was actually painless. And so what we're up to doing right now is we're scaling up the short read sequencing analysis on the full cohort. So Naomi's done roughly 1,000 patients, I think now, just because we have the data. Why not have a look and scale this up? And she's found, I think she was telling me today, 20-odd potential goers for validation. But these kind of data make me ask the question, at what point do we say, let's just do it long read to start with? And so certainly I'm starting to think about how we find the money to do that. So that's it in a nutshell. Thank you, everyone. These are a small number of our funders. But look, really appreciate the chance to talk today, and happy to answer any questions you have. Thank you.
Thank you very much, Mark. That was really eye-opening, fantastic. Any questions for Mark? By the way, Lydia managed most of that work, so I just put you in contact, but Lydia pulled all the stops to get it done. Can you tell us about cost efficiency? Because from my memory, we barcoded samples on one flow cell, so we're not doing a whole genome on one chip. It was a subset of that, right?
So I'd have to go back to our emails to find out exactly what the dollar value was, but it was actually, we did the whole thing on the one, I think, and then we might have had a makeup because we had a few samples we were really interested in following up a bit further, but it was actually really flexible. We could just say, just do these ones a bit more, and the whole thing was actually really reasonable. So I can't remember the final value, but in this project, we're dropping $2,500 in short read on a sample. So next to that, it was a rounding error, really.
No more questions for Mark? Find that. Miranda?
Thank you. Fantastic talk. I suppose with adaptive sampling, you need to know which genes you need to look at. Were there cases where you would expand your panel to say maybe it's in this gene or this gene? Were there cases where you just weren't too sure? Could you run through that?
That's a big question in the field. So the Zero really works on two sides. So number one, we do clinical, almost pseudo-clinical reporting. And that's all that I was talking about here today. And that goes back to the hospital, and we do have to be careful to only look at certain genes there because, you know, we only want to look at things that we can actually report on and are useful. So in that regard, we have an easy time of it. On the research side, though, we have probably 3 or 4 times more genes than are on the clinical list that we want to look at. So absolutely, I am thinking, when we're thinking to scale up, how can we and should we use that bigger research list? Because we're not going to find things that we don't look for. So I don't know if adaptive sequencing can do something clever like we give it the MEI sequences, or if we're just going to get a pile of rubbish, but I'd like to explore that because I'd love to be able to see things that we can't. Some of the colleagues who I work with have just said, stuff that, and Piyush was telling me today he's just got to do 20 whole genomes or 10 whole genomes at 20x anyway, just so he knows. So that's the other solution, just do the whole thing.
I think there's lots of opportunities with the adaptive sampling as well, where you can, it's kind of hard to do from a service provider's point of view, but if you're running flow cells at home, you could change the targets live. Like you start sequencing, you run a script, you see, oh, there might be something in this locus, then you can, pause the run, change the region of interest, and then zoom in there instead. So you kind of have like this hierarchical decision-making tree that you can implement. And I don't think many people are actually leveraging this capacity of this technology. Yeah, you could just, do like, I think you can go up to like 8% of the genome and you get good depletion, or enrichment rather. But more than that, you might as well just brute force it, do a whole genome.
I think we did 2-ish, wasn't it? 2%?
Yeah, it was less than 2%. We basically just pumped up their target regions to get 2% so that it would be worthwhile doing. Any other questions? No?
I'm free.
All right.
Thank you all.
Thanks, Mark. Now we're going to change gears a bit. So our next speaker is Kristina Santucci. Santucci? Yes. Is that Italian?
Yes.
Who is a PhD student?
Yes.
But you're going to be talking about your honors project.
That's right.
From Michael Janitiz lab and BABS, so local talent. I'll give you the mic and I'll put your slides up because I got to switch the PowerPoints.
Wonderful, thank you.
Just some arrows in the laser pointer.
Perfect, thank you. Hi everyone, my name's Kristina Santucci. I'm a PhD candidate here at the School of BABS at UNSW. I'm actually a first year candidate, hence why my presentation is on my honours project from last year on the discovery of novel transcriptional isoforms in the human brain. As you mentioned, I'm part of Dr. Michael Janitiz lab. We are a bioinformatics lab that looks at transcriptome profiles of various human tissues, as well as the perturbation of transcriptome profiles in various diseases, such as neurological disorders and cancer. In particular, our lab have a focus on mRNA, circular RNAs, and long non-coding RNAs. My research project for my honours focused on discovering new mRNA isoforms, and because my nanopore data was poly-A tail selected, a few polyadenylated long non-coding RNAs also did pop up in the analysis. So I should mention that the findings of my honours project was recently published in the Journal of Molecular Neuroscience. A lot of the figures and images I'm going to show you today come from that publication, as well as our team's recent literature review in Briefings in Functional Genomics for anyone that's interested. But first, a little bit of background information into RNAs. So mRNAs are generated by the splicing of precursor mRNAs. In constitutive splicing, as many of you are aware, the introns are excised and the exons are joined by splice junctions, forming the mature mRNAs. However, many alternative splicing mechanisms do exist, and this results in mRNAs with differences to the constitutive sequence, such as the skipping of an exon or retention of an intron. So from the same loci or gene, we get a variety of mRNAs, and if they are protein coding, we also find that the proteome is expanded as well because different protein isoforms are also translated. So how can we discover new RNA isoforms in sequencing data? So traditionally with short-read sequencing data, you try to collapse your reads into constitutive isoforms. This can be very challenging because, as you know, short-read sequencing requires RNA fragmentation prior to sequencing. This produces anywhere from 5 to 200 million reads with average lengths of just 100 to 150 nucleotides. So when you try to then map these reads to different isoforms, there's a lot of ambiguity. And even if you can resolve all of the different RNA isoforms coming from the same precursor mRNA, it's very hard to quantify the relative abundance of them. This is where long-read sequencing can come in to mitigate these challenges, in particular nanopore sequencing, because no RNA fragmentation or DNA fragmentation is required prior to sequencing. So you get read lengths that are up to the length of the entire transcript, which reduces this ambiguity in read to isoform assignment. So which transcript assembly tool should you use for nanopore sequencing data? If anyone has fallen down this rabbit hole, you'll know that this is actually quite a challenging question to answer, primarily because there is such a vast array of tools to choose from. As you can see in this table of the different tools suitable for novel RNA isoform detection and quantification, there's just an abundance of available tools, and even more have come out since the time of this review. Even then, multiple benchmarking analyses have come out to compare their performance. However, their findings tend to be inconsistent with one another. The long-read genome assessment annotation project recently reviewed about 14 of these tools in a very large comprehensive study. And even then, their recommendations provided multiple tools for different applications. So there wasn't a gold standard such as like Minimap for alignment. And even if you were to follow recommendations and pick one tool for your analysis, the assembly of novel isoforms are likely to differ from another tool. And hopefully I will demonstrate this in this talk today. So before I go into the method, our data that we used for this project was actually accessed from Glinos et al.'s study. He performed long-read sequencing of prize GTEx tissues. In particular, we utilised samples from the frontal cortex, cerebellar hemisphere, and putamen in the human brain. So, as I mentioned, we have these 3 brain regions. We align them to the reference genome. In particular, we actually use a 2-pass alignment. So here, rather than just using a standard tool like Minimap, which is the gold standard for long-read genome alignment, or transcriptome alignment, we also utilize a tool called 2-pass tools. So what this does is you perform your regular alignment and you supply that alignment to this machine learning algorithm called 2-pass tools, and it will score the set of splice junctions assembled in the first pass. And once it has scored that set, you supply this matrix to Minimap again in a second independent alignment for something that's called splice junction aware alignment. This essentially just filters out poor quality splice junctions and improves transcriptome assembly downstream. So once we performed alignment, we took the second pass align reads and performed transcript modelling. As I mentioned, the recommendations for what transcript modeler to use for nanopore sequencing data is a little bit up in the air in the literature. However, that long-read genome, RNA sequencing genome Assessment Annotation Project, LRGASP. They recommended for well-annotated genomes, such as humans, to use isoquant, bamboo, or FLAIR. So I decided to use all three and actually take the set of novel isoforms that were consistent in each tool. This was to reduce the chance of analysing any false positive novel isoforms, which are abundant in some of these tools. After we had our analysis set, I further filtered them out to reduce the chance of including any sequencing artifacts, such as reverse template transcriptase template switching events or intrapriming events, as well as filtering out any monoexonic transcripts. So here are the results for the transcriptome assemblies with isoquant, bamboo, and FLAIR. You can see the number of transcript models and the types of novel isoforms being constructed vastly differed between the three tools. And isoform refers to whether they were past that filtering process, if they were not monoexonic or the product of any sequencing artifacts. And I would like to just highlight the fact that FLAIR could not resolve any novel not in catalogue, novel transcripts, which means any new transcripts with new splice junctions or new splice sites weren't resolved, unfortunately. However, we still took the set of new isoforms that were consistent across these three tools for further analysis, and I'd like to go into some examples of these novel isoforms. So in total, there was 170 novel isoforms that were consistent across the three tools. They were either novel not in catalogue, meaning they had new combinations of known splice junctions or known splice sites, or we also had a few fusion transcripts and new intron retention events. When we looked at the expression of these novel isoforms in the three brain regions, we found that most were expressed in all three regions. However, we did find one that was unique to the frontal cortex and one that was unique to the cerebellar hemisphere. So for the first example, this was our novel long non-coding RNA, Bamboo TX1299. You can see its structure here in comparison to reference transcripts from the same locus. It is a novel combination of known splice junctions, and when you look at the expression of this novel isoform in the different brain regions, you can see it is unique to the cerebellar hemisphere and actually has quite a consistent and high expression in comparison to annotated transcripts of this same loci, which is functionally uncharacterized. When you compare the sequencing reads from nanopore short read sequencing and, nanopore long read sequencing and Illumina short read sequencing, you can see that this new isoform is very well covered, and the structure, although supported by the short read sequencing sample, is far clearer and more apparent with the nanopore sequencing data. As I mentioned, it's transcribed from an uncharacterized loci. This loci is antisense to a CBPL gene, which has two known antisense RNAs, AS1 and AS2 respectively. Since all annotated transcripts of this loci are long non-coding RNAs, including our new isoform, I do believe this is actually another CPVL AS antisense RNA, but this role of the gene is yet to be addressed in the literature. And as I mentioned, it showed a unique and high expression in the cerebellar hemisphere, so perhaps it regulates the expression of the CPVL gene in tissues where they're co-expressed. So for another example, we had a novel messenger RNA, Bamboo TX321. You can see the structure of this new RNA in comparison to the reference or annotated transcripts is transcribed from the gene PDLIM7, and alongside this annotated transcript, the last there, it has a increased expression in the frontal cortex and putamen, and only differs from this annotated transcript by the 5th exon. Once again, we sort of did a sanity check and looked at the aligned sequencing reads from nanopore and short read Illumina sequencing. And as you can see, considering the only difference between this new RNA isoform and its closest annotated RNA isoform match is 1 exon being the 5th, you can really clearly see that difference in the nanopore sequencing data, whereas with the Illumina short read sequencing data, you don't have that high resolution, so that read to isoform assignment is far more ambiguous with short read sequencing than it is with nanopore long read sequencing. As I mentioned, this novel RNA is messenger RNA, so it's protein coding. We wanted to see the structure of the protein it encoded and see if it had any interesting features. We performed a blast search on this open reading frame, and its closest match, unsurprisingly, was the protein translated by that similar annotated isoform. And you can clearly see the difference at that 5th exon highlighted in red. So it seems to form a truncated protein. When we put it through AlphaFold 3, the leading ab initio method for protein structural prediction, we could clearly see the structure of this protein. In particular, this first domain here was really high confidence, as well as that 4th predicted align error domain. In the middle, it wasn't so great, the prediction. However, this second domain is where the change in that 5th exon occurs, so the splice junction, the isoform is not annotated, which affects the prediction. Nevertheless, we still performed a protein domain search and a conserved region search, and we saw that the PDZ domain and PDZ and limb domain present in annotated protein isoforms were still maintained in this new protein isoform, and in fact there were the protein and peptide binding sites were still conserved, as well as 75 conserved domain hits. So to summarise, we saw that the novel messenger RNA had an elevated expression in the frontal cortex and putamen. It encoded in truncated protein due to that skipping of exon 5 relative to its closest annotated transcript match. And this was unsurprising because PD-Lim proteins have been vastly explored in the literature. They are abundantly spliced and are involved in a variety of signal transduction pathways and other biological roles such as organ development, intracellular signaling, neuronal signaling, and oncogenesis, which is functions we do suspect this RNA isoform to have because of those conserved regions. So in summary, we developed this robust analytical pipeline for novel isoform discovery. And by revisiting existing data sets, we identified 170 new transcriptional isoforms previously not resolved with short-read sequencing data. 104 of these were protein coding, 66 were long-known coding RNAs, and we showed that we could validate this isoform structure with short-read sequencing data. So I'd like to end by thanking my supervisor, Dr. Michael Janitiz, and all of the members of the Janitiz lab team, and thank you everyone for listening to my presentation.
Wow, that was fantastic. 2 papers in a couple of years of work. That's very impressive. I'm highly impressed. I have so many questions because we do a lot of this stuff as well, but I'll open it up to the floor. Do we have any questions? Thank you, Robin. At least somebody's paying attention and it's not just here for the beers.
Well, thank you for the great talk. I was paying extra attention because I was doing something similar in my lab back at home, and I wish I had heard this talk like a month earlier, because like my supervisor asked me a question in the presentation I got wrong and you essentially answered it. You had a slide at the beginning which showed the difference of long reads and short reads when it comes to looking at multiple alternative spliced exons, right? Yes, this one exactly. And you can see like exon 7, right at the end, you have like some variants where it's like included and some where it's excluded. I would imagine long reads, like the quality dropping off towards the end, right? How do you deal with that you can really be sure if the exome is like not there or if just there's a problem with the sequencing?
Thank you. So you can see if your new isoforms, which is my research focus, a full-length transcripts. This would include whether they have first up until the last exon in respect to the locus that they're transcribed from. If I flick, I actually have a supplementary slide that might aid this discussion. Yes, here. So it is trickier when you have the last or the first exon not included. Whether you call it a new RNA isoform, so I'm referring to these, they refer to as incomplete splice matches, it is hard to determine whether they are new splice or different mRNA, RNA isoforms from the same reference transcripts because you are unsure whether or not these reads are covering the entire length or are just covering a portion of these splice matches. However, I do believe that there are wet lab methods that could confirm whether or not you have the full transcript. However, I'm not familiar too strongly with wet lab approaches, but I have seen it in the literature where people can confirm whether their transcripts are full length or whether they are fragments.
Cap trap, I believe, is a method where you pull out the 5' end and sequence it, which led to the explosion of long non-coding transcripts in the latest GenCode release last year. It's one method. I think there's a few others as well. Are you happy with that answer, Robin? Did you want any more? All right. Well, while people try to express some new isoforms to answer another question, I have a very basic question about, the AI tool to filter the splicing. Do you know if that considers both the major and the minor spliceosome? Because the minor spliceosome has like a different splicing pattern.
I'm not sure. I know that 2-pass tools extract sequence features, and that's how they score all of the splice junctions present in the first pass alignment. In particular, I know that it also has a focus on resolving splice junctions near small exons, but I'm not sure whether it also includes the small spliceosome. I know it does look at every splice junction that was detected or mapped in that first pass alignment.
Perfect segue to my second question, which was microexons.
Yes.
They are common in the brain in particular. Given the, relatively high error rate, especially with indels, for nanopore sequencing, have you looked at these? Have you picked them up? Do you think it's a challenge for this technology, or is it just easy to do now?
I think so. The sequencing kit that was utilized to generate this sequencing data was quite an older kit, so I know that a lot of these challenges have recently been addressed and been updated in the newer kits. However, small exons are a problem region with at least this nanopore kit for sequencing. So the way I addressed alignment at difficult regions was one that 2-pass tools to take away that transcriptional noise and to the filtering after transcriptome assembly. However, microexons, it's likely that they might have been filtered out because it's not confident at that region, at least for the kit that was utilized in this research.
Any more questions? My last one is a comment more than a question. Flare is terrible.
I know, oh my goodness.
Yeah. The LRGASP benchmarking study, I don't think they did a very good job. They're misleading a lot of the community now.
I know, yes. nightmare to use as well as to analyze.
Yes. So have you considered maybe dropping Flare out and yes? All right.
This is my, I supervise this honours student, and he's also looking at utilising this pipeline in heart data, and we've decided to drop Flare. We're only going to look at it, because I had it, let me just go back, sorry. You can see that Flare did not resolve any genic, intergenic, or novel not in catalogue transcripts, so it significantly reduced our search space. We did, for the purpose of just comparing these three tools, still just investigated the set that was consistent among the three, but for future projects, as well as my current PhD projects, Flare is dropped. It's just going to be Bamboo and Isoquant.